The Catalyst project was about really bringing an innovation into something that can be manufactured and prepared for a clinical trial. Okay, so it was a first step to establish whether a promising idea had traction and had a good was a good fit for an eventual clinical trial. So this part was mostly done by John in his lab. So Jonathan very clearly outlined how this therapy could not only match but perhaps be superior to comparators in the field. While my role was to devise strategies to make sure that it can be implemented in a translatable way, meaning that it can be manufactured as a therapeutic cells for an eventual trial.
Multiple myeloma until recently was thought to be an incurable disease and may well still stand to be an incurable disease. But there are a number of products coming from companies like Bluebird, Nanjing Legend, that are targeting the same target that we're interested in, a target called BCMA, where they've shown very impressive clinical outcomes. Those impressive outcomes notwithstanding, there are still some serious toxicities that manifest, and our value proposition is comparable efficacy but reduced toxicity. We call it a TAC to distinguish it. The biology is different, but yes, a safer type of CAR.
Yeah, and for patients with myeloma, it's extremely important because many of these patients are older and frail. So we don't want these patients to run into massive toxicities. So this is why devising a safe product is particularly relevant here.
At the end of the day, a lot of these things are proof of principle, right? If you establish that you can do something and that infrastructure gets created, you can build on that infrastructure. So everything we're doing right now, whether it's the TAC project that Jean-Sébastien and I are working on, or it's the National CAR-T program that Vancouver and Ottawa are working on, all of these things help to establish the infrastructure, which is not just mechanical, it's also expertise. Our clinical partners have to get more familiar with these cell therapies. They're not trivial to work with. There are logistics issues in terms of shipping materials to and from manufacturing sites. There are clinical issues in terms of patient management. So the more we learn, the better able we're going to be to keep bringing these things online. And I think we all accept the fact that these cell therapies are part of the modern day cancer clinician armamentarium. And so we have to know how to use them.
And building these infrastructure is extremely important. We must not be, as a society that imports innovative cell therapies. We want to be able to use our own and perhaps even export our expertise. So, but for this, we need to establish our credibility, treat our first patients, and make sure that our innovations get through the entire pipeline of translation in Canada. There's a lot of interest for treating myeloma, especially difficult patients with innovative cell therapies. We are very well connected with our myeloma clinical colleagues, and this is something we have built on at the onset. Two well-known myeloma experts were partners on the grant, and they are questioning us repeatedly about when is this coming. So no, we are now building a case, building a group, thinking about resources to fund a first in human trial very soon. I can certainly say that there is this big gap between having a plausible product to the first patient. There's a big gap that involves money that no one wants to give, really. But BioCanRx has a program to bridge that gap, and we intend to use it as much as we can, and this will attract other partners. But it is sine qua non for success. We need to be able to treat the first patients ourselves. If we are committed to the projects and we don't rely on others to make that crucial step, things will go faster, and hopefully my patients will benefit faster.
And the infrastructure that Biocanix has created. So one of the limiting factors in these kinds of therapies is a reagent called a lentivirus. All the manufacturers worldwide are under high demand because of the success of this therapy. And so by having a capacity in Canada through Biocanix to generate lentivirus locally, that should help us to make sure we don't slip in terms of our timelines. The site where Jean-Sébastien works, Hôpital Maisonneuve-Rosemont, has an outstanding cell manufacturing expertise. Combine that with lentivirus provided by Biocanix, and that really helps us to leapfrog and make sure we move forward as quickly as we can.
The fit is excellent. Biocanix managed to bring people with different expertise and overlapping expertise. I mean, I know enough science to understand what Jonathan's working on, and then I can bridge to my clinical colleagues as well. So there's really a coherence in the expertise that's being matched through Biocanix, and that is perhaps the most important thing.
Mm-hmm.